dia.
Dari mana? Tu dah.
Sini. Sekejap. Tak tahu.
Ni buat betul sabar ni.
I'll be talking about mangrove and the factor we need to consider when to choose the right site for planting them. Mangroves are very important for our environment. However, for mangroves to thrive, we need to carefully choose where to plant them. This involves three main factors, which are location, salinity, and tidal range. The first factor is to consider is the location. Mangroves grow best in the intertidal zones, which are areas where lands meet the sea. These zones allow mangroves to interact with both land and water, creating the perfect condition for their roots to anchor and absorb nutrients. Choosing the wrong location, such as too far from inland and too far from the sea, can make it very difficult for mangrove to survive. The second factor is salinity, which refers to the salt content in the water. Mangroves are very unique because they can survive in the brackish water, which is a mix of fresh water and salt water. However, the salinity level must be just right. If the water is too salty, it will stress the mangrove and making it difficult for mangrove to grow. On the other hand, if the water is too fresh, it may not support the specific species of the mangrove that thrive in the saline environments. Understanding the salinity of in the site is important because it determines the types of mangrove we can plant and whether they will adapt successfully in the environment. The third factor is the tidal range, which is the rise and fall of the tide. Mangroves rely on the movement of tides to bring in nutrients and oxygen while removing waste. This is especially important for their root system, which are adapted to survive in waterlogged and oxygen-poor soils. As you can see in this image, mangroves have aerial roots that stick out of the ground. This will allow to plant to breathe even when the soil is submerged during high tide. Without a proper tidal range, the water may stagnate, leading to poor nutrient flow and the death of mangrove. Regular tidal flooding is essential for their growth and the overall health of the ecosystem. Therefore, when selecting a site, must ensure that it experiences natural and consistent tidal patterns. In conclusion, selecting the right site for mangrove restoration or study is critical. Key factors to consider include the location, salinity levels, and tidal range, as this determine the health and sustainability of the mangrove ecosystem. Proper site selection ensure the successful growth of mangrove, which are vital for coastal protection and biodiversity.
City lights, 도시의 불빛 아래서 dance all night. Oh yeah right, we're living life. City lights, 끝없이 all like that tonight. Let it shine. Oh yeah right, we're going up higher tonight. 전화해줘, yeah I'm on my way. Hold tight, we'll dance all day. Where the city shines so bright. know me under neon lights. Oh, 지놀자, let's just have some fun. Oh, 미루지 마라. Don't wait for anyone. 지금 이 순간에 live it up tonight. All we got's the city lights. City lights, 도시의 불빛 아래서 dance all night. Oh yeah right, we're living life. City lights, 끝없이 all like that tonight. Let it shine. Oh yeah right, we're going up higher tonight. Want to move to
So I will present to you about the data that we have obtained. For set 1, A leaf length, the mean is 10.88, the variance is 257.025, and the standard deviation is 16.032. For the leaf width, the mean is 4.38, the variance is 6.3504, and the standard deviation is 2.52. For set 2, the leaf length, the mean is 109.5, the variance is 0.4606, and the standard deviation is 0.6784. Meanwhile, for the leaf width, the mean is 3.84, the variance is 0.1249, and the standard deviation is 0.3534. Last but not least, for set 3, the leaf length, the mean is 10.37, the variance is 0.8957, and the standard deviation is 0.9464. For the leaf width, the mean is 32.80, the variance is 1.3196, and the standard deviation is 1.1487.
Let's discuss the post-lab questions for practical 1. The first question is, discuss the differences or similarity of the mean of length between different set of leaves. Do the same for the mean of width. So the difference of length between different set is the highest mean is from set 2 and the lowest mean is from set 3. The highest mean of leaf length from set 2 results from a more concentrated distribution of longer leaf length. The similarity is the range of the mean values is about 10 cm. This means that the mean value is somewhat fairly distributed among them. So, for the differences, mean of width between different set of leaves is the highest mean is from set 1 and the lowest mean is from set 3. The highest mean of length width from set 1 results from a more concentrated distribution of wider leaf length. And the similarity is the range of the mean value is in the range of 3 to 4 cm. Hence, the data is not significantly different in terms of the leaf length. Variance is the expected value of the squared deviation from the mean of a random variable. From the statement, this means that each set of leaves length and width have different values from each other and they are not fixed into the same value. Other than higher variance indicating wider distribution of values, it also means that the set of data is less consistent. Hence, the leaf size is not uniform with each other. Standard deviation is a statistic that measures the dispersion of a dataset relative to its mean and is calculated as the square root of the variance. To ensure accuracy and consistency of the growth of the leaf, it allows us to compare and replicate the results, which reduces errors and enhances the reliability of scientific research.
Now, let's discuss for the post-lab questions for practical number 2. Quadrat is an ecological sampling unit consisting of a small square area of ground within which all species of interest are noted or measurements are taken. The two basic requirements of all the quadrat techniques are the area of the quadrat must be known and the population in the quadrat must be determined exactly. For question number 3, for larger quadrat, the advantages is to reduce the sampling error. When there are more organisms in the quadrat, the data is more reliable and it is less likely to miss rare species in the small areas. Meanwhile, the disadvantages of the larger quadrat is it takes more time. Larger quadrats can take more time to be analyzed. For the smaller quadrat, the advantages is it gives off more precise data. Smaller quadrants can also detect specified pattern or interaction. And the disadvantages is it is potential for underestimation. If the organism is spread out, small quadrats may miss a lot of parts of the population, which causes the lower number of estimation. For question number 4, the issues concerning the ecologies in conducting the sampling method is mainly the size of the quadrat. The size must be suitable for a specific species to be studied. Using way smaller quadrats than the size of the species present can result in inefficient outcomes, while using a larger quadrant for a smaller species also results in unsuitable outcomes and miss the finer scale patterns that are crucial to observe. Last question, the distribution of the plant species in the quadrat is a random distribution. Random distribution occurs when the position of each individual in a population is independent of the others. There are several factors that may have contributed to this type of distribution, such as first, environmental conditions. Second is the random seed distribution, and the last one is human factor.